So I'll introduce Dr. Lucas Cheadle. He did his doctoral training at Yale, and then afterwards was a postdoc in the lab of Dr. Michael Greenberg at Harvard Medical School, and currently he is an assistant professor at called Spring Harbor Laboratory. I'm looking forward to your talk, Lucas.
Great. So let me just share my screen here. And you guys seeing my main screen or my presenter view?
We see your main screen.
Main screen, yeah.
Okay, well, I just want to say thank you very much for including me in this symposium. I'm clearly aware of all of the work from the folks that are presenting here, and I actually think that we sort of make up this core group of people who are really starting to move the field of microglial research into a new direction, which is to think about all of the fantastic roles they play even beyond the earliest stages of synapse development. And I also want to thank the organizers, because obviously you guys have recently put out really informative papers that have really shaped sort of where this new line of investigation is going for me. So it's a real honor to be here. So I'm going to be discussing a paper that came out online in September from my postdoc, in which we look at a role for microglia in sensory dependent synaptic refinement. And I'm not going to go, figure 1, figure 2, figure 3. I just wanted to give a little bit of a more developed sort of idea of the work so that I could just focus on what I think are the most interesting parts, just given the time limit. So as organisms navigate the natural world, we're constantly sampling our environment for information to help us to determine, for example, what to eat or when to sleep or where to hide from dangerous predators. And this information from the environment interfaces with the brain in the form of sensory experience. And the ability of the brain to interpret and respond to sensory stimuli is an important aspect that determines animal behavior and ultimately is paramount to animal survival. One of the most important roles that sensory experience plays in the brain is to regulate a process that occurs during postnatal development, and that's called synaptic refinement. Early on in the development of the brain, synapses initially form a very high number of synaptic connections with one another. But many of those connections are actually eliminated in postnatal development in a process that's called synaptic refinement. So synaptic refinement really refers to this balance between the strengthening of a small number of synapses and the elimination of others. And that process is really beautifully illustrated in these Golgi-stained sections of human cortex, which demonstrate the robust elaboration of synaptic connectivity between birth and about 6 years of age, followed by what you can see as a significant pruning down of connectivity that becomes apparent around the time point of adolescence. And this pruning down of synaptic connectivity is really critical for establishing the efficient and precise connectivity of the mature brain. But while we know quite a bit about the molecular mechanisms that underlie this early phase of synapse assembly, we know much less about the fundamental mechanisms that guide synapse elimination, particularly the phase of synapse elimination that occurs in direct response to sensory experience. So one of the systems that's been utilized to study this process of synaptic refinement is the retinogeniculate circuit of the mouse. And this is the connection between the axons of retinal ganglion cells in the eye and their postsynaptic targets, the relay neurons of the dorsal lateral geniculate nucleus of the thalamus. And I think most people who have done any kind of reading on microglia know that this has been a really critical system for establishing some of the roles of microglia play during normal brain development. So what's known about this system is that at birth, it's roughly established, but it's refined over the first month of postnatal life. And that refinement period begins with a pre-sensory phase during which microglia remove excess synapses through phagocytic engulfment. So this is sort of similar to what Ashley was just talking about, except here we're talking about the microglia removing synapses, not OPCs. But what was discovered by Ben Barris, Beth Stevens, and Dory Schaefer and others, is that microglia tend to not engulf synapses following the onset of sensory experience at eye opening. And so this has led to a little bit of a mystery as to whether microglia actually play an important role in the sensory dependent phase of refinement in the retinogeniculate circuit, or whether their roles are really more sort of restricted to these earlier phases of development. And so this sort of understanding how sensory experience regulates synaptic refinement was a major focus of my, my postdoctoral work. And so the first thing we did was we actually had a previous paper where we showed that sensory experience is important for upregulating a cytokine receptor called FN14 in relay neurons of the DLGN. And we found that that receptor is really important for refinement. So in the newer paper that we're discussing today, I'm going to tell you about some other mechanisms that we found converge upon FN14 to mediate refinement. And so I just want to give you a brief background of what FN14 does in the brain, most of which we published in 2018. But this is also a summary of the first couple figures of the newer paper. And what we found is that if we looked at retinogeniculate connectivity in a constitutive FN14 knockout mouse, we saw that the relay neurons actually maintained an excess number of synapses coming from the retina compared to a wild type, which indicates that there's that synapse elimination does not occur appropriately in the absence of FN14. And we dug deeper into that in the new paper by looking at synaptic structure in these knockout mice. And what we found is that FN14 knockout mice actually contained a lower number of synapses, spine-associated synapses, dendritic spine-associated synapses than their wild-type counterparts. And we think that this decrease in the number of synapses connecting the retina to the DLGN in an FN14 knockout is what leads to the inappropriate or deficiencies in synaptic strengthening in response to experience. Now, consistent with this process actually requiring visual experience, when we dark reared a wild-type mouse, we found that there was a strikingly similar physiological phenotype to the FN14 knockout. And when we looked at structure, we found a consistent phenotype there as well. And so the bottom line is that we've identified FN14 as a molecule that's upregulated in relay neurons of the DLGN in an experience-dependent manner that goes on to balance both synaptic strengthening and synapse elimination. So one of the main themes of the newer paper that we published is we wanted to look at whether the cytokine ligand of FN14, which is called TWEAK, could be involved in this process of experience-dependent circuit remodeling. Now, just to give you an idea of what's known about this cytokine signaling pathway outside of the brain, where it's been much more extensively studied, it's been shown that FN14 expression tends to be relatively low in healthy tissues, but that upon injury or pathology, tissues can actually drastically upregulate the expression of FN14. Now, it's known in the field of immunology that at sites of injury or insult, macrophages can infiltrate where they can perform their phagocytic functions and clear up apoptotic and damaged tissue. Well, macrophages tend to be really high expressors of the ligand for FN14, which is a tumor necrosis factor cytokine called TWEAK. When they found that cytokine TWEAK binds FN14 in these tissues to remodel the tissue, for example, to establish fibrosis. And so we were really curious about whether TWEAK may play some analogous role in this process. So the first way we looked at that was to take advantage of a single cell transcriptomic resource that we had recently published, where we looked at gene expression in all the major cell types of the DLGN across postnatal development. So when we specifically look at TWEAK transcription in the data set, we find that TWEAK is relatively lowly expressed in a number of different cell types before eye opening, which is around the time of P5 and P10. But that right around the time of eye opening, TWEAK becomes highly upregulated in microglia specifically, and its expression continues to increase in microglia up into the period of the sensory dependent critical period of refinement, which begins at P20. And so this is actually quite an interesting expression pattern because we know TWEAK is highly expressed in macrophages of the periphery. This really makes sense given that microglia are highly specialized macrophages. But what's actually striking here is this sort of notion that TWEAK is increasing along with eye opening, which led us to hypothesize that visual experience may actually transcriptionally induce TWEAK in microglial cells. And so to test that, we moved into a visual stimulation paradigm that we developed in which we can rear mice in normal conditions up until they're about P20, postnatal day 20. And then we put them in complete darkness. We rear them in darkness for about a week until P27. And the reason we stop at P27 is this is really the peak of sensory dependent refinement in the DLGN. So after this visual deprivation, we can take the tissue of the mouse and consider that our unstimulated control, or we can re-expose the mouse acutely to light, for example, for 8 hours is what I'll be showing you here. And we found in previous studies that that's really a nicely strong way to induce activity-dependent gene expression in neurons. And so we performed this analysis, and then we performed fluorescence in situ hybridizations to look at TWEAK expression. So what I hope you can see in these confocal images is that in a dark-red mouse, microglia, which I'm showing here using a marker for fractalkine receptor, in a dark-red mouse, we see very little TWEAK expression, if at all. But when we look at TWEAK expression in a light-red mouse, we see a significant increase in the amount of TWEAK expressed by microglia, consistent with visual experience playing an important role in upregulating TWEAK expression selectively in microglia. And when we quantify this by measuring the percentage of microglia that express appreciable levels of TWEAK in a light-reared mouse versus a dark-reared mouse, we see again this really significant increase in TWEAK expression in light-reared mice. There's something else to sort of mention here that I think is equally interesting to that is that there's actually heterogeneity within this response. So some microglia induce TWEAK expression in response to light stimulation while others don't, which indicates a potential functional heterogeneity of microglia in the DLGN, which is something that we're very interested in as a lab and exploring now. But the fact that we see this TWEAK expression in microglia around experience-dependent refinement was very exciting because it indicated to us that maybe TWEAK is binding FN14 to regulate synapse refinement in response to experience. And so the first thought we had was, well, perhaps TWEAK is directing microglia to engulf synapses. And so the first set of experiments we did was simply to look at phagocytic engulfment of synapses in a constitutive TWEAK knockout mouse. And we looked at this in a number of different ways. And here what I'm showing you is an example of what was actually really nicely already introduced by Ashley, so I won't go into the details, but it's a way that we can look at synaptic material inside of volumetric reconstructions of microglia. When we analyze synaptic engulfment in mice lacking TWEAK at an early time point, P7, and a later time point, P27, these analyses revealed that TWEAK is dispensable for phagocytic engulfment. So the next question, maybe A, was TWEAK actually involved in this synaptic remodeling through FN14? And if so, what other mechanism beyond phagocytic engulfment could be involved? And since we had established this really robust role for FN14 in maintaining the correct number of spine-associated synapses, we next looked at this parameter in the knockout mice using Golgi staining. So just as a reminder, what we saw when we removed FN14, and this is actually, these data are from a conditional knockout mice where we actually removed FN14 selectively from relay cells. What we see when we remove FN14 selectively from relay cells compared to a control is a significant decrease in the number of spines. We think this is corresponding with an inability of particular synapses to strengthen. When we look at a wild-type dark-haired mouse, we see this exact same effect. So this is, again, consistent with the fact that FN14 functions in an experience-dependent mechanism. But our next hypothesis was that in a TWEAK knockout, we would see some sort of similar effect, which would sort of highlight that TWEAK and FN14 may be functioning in a straightforward manner as a signaling pathway to establish synapses. But what we actually found was that there was a significant increase in the number of synapses in mice that were constitutively lacking TWEAK. And we think that this is a real effective loss of TWEAK on spines because we can rescue this effect by virally overexpressing TWEAK in the DLGNs of a knockout mouse. And so this was really not what we were expecting, but it allowed us to establish a model for how TWEAK and FN14 might be regulating synapses. So our model states that in normal developmental context, visual experience upregulates FN14, which is a synapse-associated protein that leads to an increase in the number of dendritic spines and an increase in the number of synapses on spines to drive the synaptic strengthening and maintenance of a subset of retinogeniculate synapses. But at particular synapses that may be specifically nearby or proximal to microglia that induce TWEAK, we reason that TWEAK could bind to FN14, leading to a decrease in the number of spines and synapses, and ultimately synapse elimination. And one of the things that I think is really interesting about this model is that it really starts to explain this balance of refinement, right? I told you in the beginning of the talk that refinement isn't just about synapse elimination. It's about finding that balance between which synapses are selectively strengthened and maintained and which synapses are eliminated toward that goal. Now, what our model starts to do, which I think is really important, is to establish a way through which a single protein, in this case FN14, expressed in a given neuron, may go on to lead to the strengthening of some of that neuron's synapses and the elimination of others. And if it's true that FN14 functions in this way, then it establishes an interesting role for extracellular signals coming from glial cells in sort of regulating that important process. And so we came up with a number of hypotheses for what would happen if we virally overexpress TWEAK in the DLGN. And our first hypothesis was that if we overexpress TWEAK in the DLGN, it would bind its receptor FN14 and decrease the number of dendritic spines. And so that's exactly what we saw when we virally overexpressed TWEAK in the DLGN. And the second prediction that we made was that because sensory experience is important for FN14 expression, if we overexpress TWEAK in the DLGN and then we put the mouse in the dark, we might not see an ability of TWEAK to decrease spines further. And so that's what we see here. And as I told you before, we had noted that darkering a wild type mouse under control conditions leads to a decrease in spines. We were able to independently repeat that result here. But now we see that TWEAK is no longer capable of decreasing spine number. And finally, we hypothesized that in the complete absence of FN14, which we achieved by crossing our conditional knockout mouse to a vGlut2 Cree driver, we reasoned that TWEAK would no longer be capable of decreasing dendritic spines in this case either. And of course, that's what we saw. And so we think that these data really establish an important role for TWEAK FN14 signaling in synapse elimination. But one question I haven't addressed is, I haven't really convinced you, probably, that microglia are the relevant expressors of TWEAK to mediate this process. We think they're the major expressors, but we don't necessarily know that it's microglia-derived TWEAK that's performing these functions. And so to address that question, we developed a conditional knockout mouse where we've actually flocked the third coding exon of TWEAK. And we crossed this to a microglial Cree driver. In this case, we just used the CX3CR1 Cree. And what we found was that we saw significantly more dendritic spines in these Cree-positive mice versus Cree-negative controls. And that's consistent with microglia specifically being necessary for dismantling these synapses in the context of sensory-dependent refinement. And in unpublished studies, we've actually also looked at spines in the DLGNs of mice where we've depleted microglia selectively during this experience-dependent phase of refinement. And we've seen, again, a significant increase in the number of spines are left when we remove microglia. So we actually think that this sort of highlights that TWEAK is maybe a single component of a larger molecular repertoire engaged by microglia to remodel developing synapses or to lead to their disassembly. So if microglia express TWEAK, but TWEAK is not necessarily important for engulfment, how might TWEAK be regulating the synapse disassembly? Since I told you previously that we found that not every microglial cell expresses TWEAK in response to experience, there's really heterogeneity in this aspect. We next asked whether the microglia that do tend to express TWEAK in response to experience might actually be more proximal, proximal to fewer synapses than those that don't induce TWEAK. And the reason we think that is because TWEAK is a secreted cytokine. So we reason that the synapses near microglia expressing it may actually be preferentially disassembled. And so to look at that, we adapted the engulfment assay that was developed by Dory Schaefer when she was in Beth Stevens' lab. And that was already really nicely introduced by Ashley. And now what we do is we combine fluorescence in situ hybridizations so that we can look at TWEAK expression at the RNA level within a microglial cell, in which we can also look at synaptic engulfment by staining with an antibody, in this case, an antibody to VGLUT2, which stains retinal inputs. And this is just an example of a 3D representation of a microglia in green, containing that TWEAK RNA in red and synaptic inputs in blue. And I think that it's a really nice technique that allows us to look one microglia at a time in a way that's really, I think, complementary to single cell transcriptomics, because it doesn't involve having to remove the microglia from the brain and disassociate them, potentially complicating how we read out the answer. So, so we did this analysis, and this is just an example of what some of the images looked like whenever we stained for microglial markers, probed for TWEAK mRNA, and also stained for retinal inputs. We can use Imaris, a fantastic software package for imaging analysis, to reconstruct individual microglia. And that allows us to really confidently say which of these many different puncta are actually contained within our microglial cell. We can threshold for the number of retinal inputs that we're looking at, and we can then ask how many of these retinal inputs are inside of this particular microglia. So we can do this in the same DLGN essentially, but choosing a high TWEAK expressor or a low TWEAK expressor, and comparing the number of proximal inputs that are particularly nearby microglia. And I should say that this engulfment assay is really helpful, but it's not perfect in every way. It's the diffraction limit of light with microscopy tends to be about 200 nanometers, so it's certainly possible to think something's engulfed or something's nearby when it's not really touching. But at the same time, I think if we're consistent across conditions, we can still learn something about how microglia may interact with synapses through this assay. And indeed, if I were to zoom in on some of these volumetric reconstructions, I hope that you can appreciate that it looks to us like higher TWEAK expressors tend to be more proximal to retinogeniculate inputs than microglia. Sorry, it's the opposite. They have fewer proximal inputs compared to microglia that tend to express lower levels of TWEAK. And we quantified this first by just asking whether there's any type of correlation between these two factors, the amount of TWEAK expressed by a microglia or the number of inputs that are proximal. And indeed, we did find a significant negative correlation between these two factors. And I think it's actually a little bit easier to visualize if you look at it in a bar graph where the y-axis represents the number of inputs nearby, and the x-axis represents increasing bins of TWEAK volume. So just to summarize the results of the study, we extended what's known about the roles of microglia in synaptic refinement from presensory phases into sensory-dependent phases of retinogeniculate refinement. And what we show is that sensory experience is important for transcriptionally upregulating the receptor FN14 in neurons and its ligand TWEAK in a subset of microglia. We find that TWEAK releases microglia to provide a local signal to synapses that disassembles and eliminates them, while microglia that don't express TWEAK in response to experience tend to be nearby synapses that remain healthy and stable. And so I really think the two major take-home messages from the paper are that we've now discovered a new mechanism of sensory-dependent refinement that involves microglia but is distinct from synaptic engulfment. And second, we've identified an important role for microglia in an important way in which their transcriptional profiles can be modified by cues from the external world, which I think is really interesting given their identities as immune cells. And so with that, I'll thank all the folks that I worked on this project with as a postdoctoral fellow. And in ongoing work where we're following up on this, I want to thank the members of my lab now at Cold Spring Harbor, Johan, Uma, and Austin, who are really a fantastic team. And we're generally open. If there are any trainees out there thinking about doing a postdoctoral fellowship, please feel free to email me.
Very nice talk, Lucas. So I have a question first off. So do you think that FN14 would mainly promote the formation or just the maintenance of spines?
Okay, so the question is, does it support them? So I'll tell you this. At P20, there's no difference in the number of spines in an FN14 knockout mouse. So the question of, so there's no, so I guess I would say I think it's probably a little bit of both, because in the P20 mouse, there's the same number of synapses. And in the P27 mouse, we see that the number of synapses in the wild type bulbous synapses doesn't necessarily change much between P20 and P27. And so I guess I would say that it's probably a formation phenotype, but we're really not sure. So one of the things we're doing to follow up on this work is you probably caught on that everything is in fixed tissue. So it's difficult for us to make comments about what's happening actively. So now we're using 2-photon microscopy to actually watch what this non-phagocytic signaling looks like in terms of deconstructing spines. So I think we'll have a better clue for what's really going on soon.
Cool. All right. So there's a question. Have you observed heterogeneity of TWEAK expression throughout the CNS or only within the DLGN?
Well, if we look at some of our other data sets, it actually does look like TWEAK expression is expressed in a number of different cell types across the brain. It's interesting for us, FN14 in the LGN is so robustly selective to relay neurons that was very clear, but FN14 is expressed in other cell types once you get outside of the DLGN. TWEAK in the DLGN, on the other hand, was most highly and inducibly expressed in microglia, but we also did see lower levels of TWEAK in other cell types like endothelial cells, which is one of the reasons why we had to do the conditional knockout experiment. But I think if you look at TWEAK in other parts of the brain, it's also diverse. So definitely heterogeneous, could be playing similar roles or other roles in the rest of the brain, and we're just not sure yet.
All right. We have time for one more question. Can you comment on other types of excitatory synapses in the DLGN and whether their refinement might be regulated through similar or different mechanisms?
So the other major excitatory synapse type in the LGN is corticothalamic input, which is VGLUT1 positive, and actually makes up the bulk of the synaptic inputs to the DLGN. Even though the retinal inputs are stronger, they're fewer, if that makes sense. So we don't actually see any differences in VGLUT1 corticothalamic synapse development in the absence of FN14. So we didn't really move forward with that, looking at that as a possible role for TWEAK that may be independently of FN14, but we don't think it's a similar mechanism simply because FN14 is dispensable for it, whereas it's required for the retinal refinement.
Okay. Thanks a lot,
Thanks.
Lucas.